Hello everyone, and welcome back to the lecture series on the physical properties of solutions. In the previous lecture, you were introduced to colligative properties of solutions, which depend on the number, but not the type, of dissolved particles. The colligative properties discussed were vapor pressure lowering, boiling point elevation, freezing point depression, and osmotic pressure. Perhaps the most important colligative property for biomedical students is osmosis, which is the flow of solvent through a semi-permeable membrane from a more dilute solution to a more concentrated one. In systems like the one presented here and in living cells, the semi-permeable membrane allows solvent molecules but not solute particles to pass through. You learned that osmotic pressure is the pressure required to prevent osmosis from occurring. In this lecture, we will continue our examination of colligative properties. You probably know that human blood consists of red blood cells, erythrocytes, white blood cells, leukocytes, and platelets, thrombocytes, suspended in plasma, which is an aqueous solution containing a variety of solutes, including salts and proteins. Each red blood cell is surrounded by a protective, semi-permeable membrane. Inside this membrane, the concentration of dissolved substances is about 0.3 molar. Similarly, the concentration of dissolved substances in plasma is also about 0.3 molar. Having the same concentration, and therefore the same osmotic pressure, inside and outside the red blood cell prevents a net movement of water into or out of the cell through the semi-permeable membrane. To maintain this balance of osmotic pressure, fluids that are given intravenously must be isotonic to plasma. Thus, 5% dextrose and normal saline, which is a 0.9% sodium chloride solution, are two of the most commonly used isotonic intravenous fluids. A solution with a lower concentration of dissolved substances than plasma is said to be hypotonic to plasma. If a significant volume of pure water were administered intravenously, it would dilute the plasma, lowering its concentration and making it hypotonic to the solution inside the red blood cells. If this were to happen, water would enter the red blood cells via osmosis. The cells would swell and could potentially burst, a process called hemolysis. On the other hand, if red blood cells were placed in a solution with a higher concentration of dissolved substances than plasma, water would leave the cells via osmosis. Such solutions are said to be hypertonic to plasma. In this case, the cells would shrink, a process called crenation, which is also potentially dangerous. Accordingly, the osmotic pressure of human plasma must be maintained within a very narrow range to prevent damage to red blood cells. Hypotonic and hypertonic solutions can be administered intravenously to treat specific medical conditions, but the patient must be carefully monitored throughout the treatment. Interestingly, humans have for centuries exploited the sensitivity of cells to osmotic pressure. The process of curing meat with salt or sugar causes crenation of the bacterial cells that would otherwise cause spoilage. The phenomenon of osmosis has found another important application in what we call hemodialysis. We know that the function of the kidneys is to filter metabolic waste products such as urea, toxins, excess mineral salts, and water from the blood. Therefore, kidney malfunction can be life-threatening. When the kidneys fail, the blood must be purified through a dialysis procedure in which waste products are removed from the body by artificial means. Specifically, in hemodialysis, blood is removed from a body and pumped into a machine that filters out toxic substances and then returns the purified blood to the patient's bloodstream. Inside the dialyzer, an artificial porous membrane separates the blood from a fluid called dialysate, which contains many of the blood's components at similar concentrations to prevent any net movement of these components between the blood and the dialysate through the membrane. The size of the membrane pores allows only small waste products to pass through into the dialysate to be washed away. Meanwhile, proteins, cells, and other important blood components are too large to pass through the membrane and remain in the purified blood. Typically, a hemodialysis procedure lasts 4 to 6 hours, and in many cases, 3 weekly treatments are required to purify the blood. In the end, let me present some applications of osmosis in ophthalmology. Osmotic agents, such as mannitol, are used in the treatment of acute glaucoma. Osmosis is used to reduce the volume of the vitreous humor, the gel-like substance in the eye, which increases the intraocular pressure. Osmotic agents like mannitol, administered intravenously or orally, draw water out of the eye, reducing the pressure and relieving symptoms of glaucoma. The osmolarity of eye drops is also crucial to prevent damage to the delicate structures of the eye. If the osmolarity of eye drops is too high or too low compared to that of the natural tear film, it can lead to discomfort, irritation, or even damage to the ocular tissues. Maintaining an optimal osmolarity is key to the safety and effectiveness of treatments like lubricating drops, antibiotics, or anti-inflammatory medications. Finally, when patients undergo cataract surgery and receive lens implants, the solution used for irrigation during the surgery must maintain osmotic balance to prevent swelling or damage to the eye tissues. Osmotic pressure is also considered when designing intraocular lenses to ensure that they do not interact adversely with the surrounding tissues. To sum up, in this lecture, we revisited the colligative properties of solutions and explored some of their applications. You should remember that two solutions with the same osmotic pressure are called isotonic. Hypotonic refers to a solution with lower osmotic pressure, while hypertonic refers to a solution with higher osmotic pressure. These terms are often used in reference to human plasma, which has an osmotic pressure of 7.6 atmospheres. We briefly discussed the importance of osmosis in the treatment of eye disorders. In the following module, I will introduce you to the main types of chemical transformations that occur in aqueous solutions, which will help you build a general understanding of acids and bases. Thank you for your attention and participation.